Welcome to our presentation on improving management of ex situ plant conservation. My co-presenter and I will be breaking this presentation into 3 bits. I will start with a bit of background on ex situ collections and some of the genetic concerns we have in maintaining a genetic collection. Seana Walsh will follow with an example of a species where we have modified our management plans based on genetic concerns. And then finally, I will finish off with some future directions. To begin with, I should introduce myself. My name is Jeremie Fant. I'm an electoral ecologist at the Chicago Botanic Gardens, and my co-presenter is Seana Walsh, conservation biologist at the National Tropical Botanic Gardens. To start the presentation, I thought I'd go into a background on how collections have come to be. Historically, collections have been done with the aim of collecting useful plants. So aside from agriculture, there's famous examples such as the Hanging Gardens of Babylon, where plants were collected for aesthetic beauty. And even before that, there is a famous example of an emperor in China who collected plants for medicinal use, which he collected with the idea of creating more community health. Although collections can be dated back many centuries, the ideas that we think about for botanic gardens and collections can really date back to Padua in 16th century, which was one of the first botanic gardens where collections were made with a sort of a systematic approach of collecting plants. These sort of botanic gardens spread throughout the world, particularly throughout Europe, although in many European countries, these botanic gardens were more about demonstrating colonial power and colonial collections than really with the idea of conserving population. What is important to remember is that many of these collections had very different focus. Some collections were made simply with the idea of saving plants of usefulness, herbal plants or plants of medicinal use, or sometimes they were just mainly collecting things that some people found interesting or fascinating, or were oddities that impressed people. However, it's only recently that now collections have turned from the idea of demonstrating sort of a wealth of interesting and fascinating species to one where conservation has become the focus. And now many botanic gardens are shifting focus of turning their collections from one which was based on oddities and displays to ones where conservation becomes a central focus. With this shift from collections based on conservation, one thing we need to be aware of is that conservation can mean different things to different people. When we talk about biodiversity, we actually include a multitude of different types of diversity. We include everything from within species diversity, between species diversity, and even ecosystems diversity. And so when a collection is built, quite often the focus has been species diversity, with the idea of collecting one example of every species. The problem with this sort of focus is that it lacks any sort of significant genetic diversity and therefore can limit its usefulness for conservation. Although this is changing, and so there are now many examples of collections that have been started to be made in the last 2 decades, where we're starting to see genetic diversity being really considered in collections. And so now data on botanic gardens around the world, so there's roughly 278 seed banks around the world, 133 tissue culture facilities, 326 conservation programs that exist, and 199 botanic gardens are involved in reintroductions. The national collection here at CPC has over 788 native rare species being conserved with the idea of conserving genetic diversity. The ways in which collections are being made can vary from institution to institution, and their value for genetic diversity and their longevity and cost will vary depending on the focus. So there are I've broken up this into 2 types of collections that are really a focus. One type is a sort of a germplasm focus, where we collect samples which are held in some sort of stasis, which allows them to be preserved for a very long time. Often this sort of germplasm collection will allow you to collect many individuals at a relatively low cost. And this is in contrast with a living collection, where plants are maintained and allowed to flower and reproduce and mature as they would normally. And this has some limitations, particularly on how many individuals you can hold and how long you can hold them for. However, when we look at the problem from just the perspective of threatened or endangered species, we can see that we're not always doing a great job of making sure that these species are in our collections. With vulnerable species, only 36% of vulnerable species are currently in any collections, either germplasm or living. And the numbers are even worse for imperiled and critically imperiled species. When these endangered species do enter into collections, we see that in many cases, unfortunately, most of these plants are maintained in a very few number of collections. So there is limited redundancy in our collections, with the exception of a few examples of some species which seem to be found in multiple collections around the world. One of the reasons for this is that the most commonly form of conservation of collections is seed banking. Plants, thankfully, many of them produce seeds which allow them to remain in a state of stasis and can be preserved in different conditions, anything from freezing to drying, which allows those many individuals to be stored at the seed stage. Unfortunately, not all species can be seed banked, and so this is not an option for everyone. And in those plants, their options are limited to either a living collection or something like cryopreservation or tissue culture. When the plant is forced to be represented in a living collection, this presents us with a lot of challenges which aren't present in the case of a seed bank. Once we have a collection, we really will have what is effectively a very small population of individuals. Those individuals then need to be maintained for multiple generations, which increases the chance of adaptation. And then often these populations are made with very few limited number of founders. And overall, together, these create a challenge for conserving genetic diversity. The main challenges that these collections face are the same challenges that are faced by any small population. Richard Frankham famously published a survey which showed that the three main issues of extinction within small populations are mutation accumulation, loss of genetic diversity, and inbreeding depression. And those three genetic processes are often associated with extinction of populations, but also can be associated with extinction of our collection. Mutation accumulation is an interesting example and is one we often ignore when we're talking about restoration, simply because mutation accumulation requires many generations and is often something we not usually have to worry about within our species and is more commonly seen in bacteria and viruses and those sort of species. However, the reality is in cultivation, we do propagate a lot of species clonally, and it's quite often seen when plants are clonally reproduced that they do accumulate viruses and other mutations over time, and these have produced many horticultural varieties, but also could be a problem for many of our species. It also highlights a very interesting problem that we have for collections, and that is that we do rely on things like vegetative propagation, and vegetative propagation can create a situation where the number of individuals we have doesn't really reflect the number of genetically unique individuals that we need in our population. The importance of this distinction can be best reflected in the extinction vortex. The extinction vortex is the idea that there are many processes ongoing which, in accumulation, lead to an increased risk of extinction. One aspect of the extinction vortex is demographics, and so demographics are really mostly reflecting the number of individuals you have in that population, and that can be best represented by just a count of the number of individuals in your population. However, the genetic processes that are ongoing in this extinction vortex, such as inbreeding and loss of genetic diversity, really come down to effective population size. So it is the number of unique individuals that are contributing to that population. And so these differences are often sometimes the same thing. So there is a little difference between N and NE, but in some cases, particularly when you have clonal plants, you can see an increasing difference between the sensor size and the number of genetically unique individuals. The other issue that Frankham identified was the loss of genetic diversity. Now, this is assuming that genetic diversity was collected from the beginning, but as I pointed out previously, many collections focus on one representative per species, and so do not always reflect the full complement of diversity that can be found in the wild. And these images of potatoes demonstrate the difference between collecting a single individual or a different single representative of a species versus the whole breadth of species diversity that can be out there. Now, in this image, I have obviously focused on phenotypic diversity, which is very obvious to us, but I will remind people that not all diversity is visual and stuff that we can see, and most of it is well hidden. If you think about wolves, we know well that wolves are the common ancestor to what is most of the dog species that we have, or dog varieties that we have in our homes. However, if you were to look at a band of wolves, you probably would not believe that they have the capacity to produce all this diversity, and that most of it is hidden, or at least not currently present. So when we're trying to save genetic diversity, we are trying to save the potential to produce all the diversity that we need, or that could be needed in the future with climate change and those sort of situations. The third and the final issue that Franken brought up was inbreeding, and particularly inbreeding depression. I know many species of plants inbreed frequently on their own and do this naturally, but we do know that inbreeding over a long term can cause consequences for populations. And this is something that has been recognized way before, even before Darwin's time. We knew that inbreeding was not a good thing. And so here in most collections, we see that what happens with crosses or crosses between individuals is it's really done by opportunistically. So if we have pollen from one plant, we just use it to pollinate plant from the next to it. And quite often there is no consideration on the relatedness of plants in collections. And here we have an example of Amorphous titanium. It's a classic example of you only get one flower from a plant and they flower very infrequently and there's very little overlap in flowering. And so just by chance alone, there's a very good chance that you're going to be mating between two related individuals. And here is an example of what happens when you do that. Many of the plants that are produced can be, if sufficiently inbred, achlorotic and therefore not producing chlorophyll. And this sort of problem is very common and has been identified in zoos for a long period. So pedigrees were recently done of the golden lion tamarin. I say recently, it's a couple of decades. Even though they were able to generate hundreds of individuals across the world, when someone finally took the effort to put a pedigree together of how each individual was related to each other, what they found was very surprising, that actually most of the individuals within the collection were derived from three very fecund individuals, and particularly two mothers which were very good at raising youngs. And so without actually intentionally doing anything and just using the best breeders and the most fecund individuals, they created a bottleneck, which resulted in a lot of genetic diversity lost and a lot of inbreeding unintentionally wise. Now, why do we care about this? We care about it because we know that inbreeding increases the chance of mortality, and it has been shown over and over again that as inbreeding increases, the chance of mortality will increase, and this has been shown in plants and animals for many species out there. So with that, I'd like to just remind people or summarize that there are a number of issues that are happening in collections which are unintentional but have important genetic consequences. The first and the most, I think one of the most important, is that people need to recognize the difference between the number of individuals you have versus the number of individuals that you started with. And this is where founders become an important concept. How many individuals were brought into collections really reflects the genetic diversity of your collection. Also, we need to recognize that genetic diversity is not created. Once you brought genetic diversity in, waiting for mutations to create diversity is a waste of time. So you only can have as much diversity as you brought in, and under most circumstances, we can expect to lose diversity rather than gain or even maintain diversity. In collections, because we have small populations, natural pollination may not always be the best solution. Without tracking who you're mating between, you could inadvertently be inbreeding, or even worse, selfing plants, which is ultimately just going to accelerate the problems of inbreeding. And then also we need to recognize that not all individuals are equally valuable. And I point this out because often we make collections of seeds from a plant, and although we can collect hundreds of seeds from an individual, we have to recognize that all those seeds represent just siblings of one collection and do not actually represent hundreds of unique individuals. So just recognizing even the fact that when we make a seed collection, we are only collecting from one maternal line and a restricted amount of genetic diversity is an important thing to consider when we were making our collection. Many of these issues we are facing with our living collections are the same issues that are faced by the zoo community. In the last 3 decades, the zoo community has seen a shift in the way they manage their living collections. More specifically, they looked at the idea of managing their collections as a meta collection rather than each institution managing their collections on their own. This has required things like in creating a stud book to manage the pedigrees of every individual that is known in collections, and then also to use management software to help them make decisions that retain genetic diversity and minimize inbreeding. This level of detail is well represented by this photo I took when I was at San Diego Zoo, where this scientist was looking for any evidence of any eggs in a recently deceased rhinoceros, with the idea that even though this individual has died, that her genetics could potentially be preserved in cryopreservation and allow her to be continually used in collections for perpetuity as long as we have eggs from this individual. This sort of level of detail of saving every genetically valuable individual in a collection is one that we in the plant community could probably face as well. The potential of a genetic-focused collection management scheme can be best reflected in what I consider one of conservation's best success stories. The black-footed ferret was at one point considered extinct, with no population seen for a number of years, until 18 individuals were found on a farm in Wyoming. The Fish and Wildlife Service went in and collected every one of those 18 individuals, and although they were all from a single colony, they were quickly genotyped and then identified relatedness between individuals, and then a breeding program was begun. That breeding program was distributed over 6 institutions, and each institution was working towards increasing the number of individuals. From there, we've seen that more than 7,000 black-footed ferret kits have been born. From those 7,000 individuals, 2,600 of these animals have been introduced into the wild, and 8 populations are currently considered to be self-sustaining. This was managed in less than 3 decades. And although the money that they have is much greater than any plant community could expect, we do still see an example where 18 individuals from one colony were quickly turned into 10 restorations and spread throughout the country, increasing the likelihood that this species will be persisting into the future. So we as a team have got together to try and see how this same sort of management protocol could be used in the protection of many endangered and threatened plant species. And there are a number of people that we should thank, but we don't have time. But this is a collaborative effort between many different individuals. The first lessons that we learned from the zoo was don't put all your eggs in the one basket, and multiple institutions are important if we're going to safeguard genetic diversity, especially with drift. We need to keep track of founders and their genetics, and less concentration on the number of individuals we have. We need to do our best to avoid inbreeding, and where possible, know the genetic origin of any individuals, and that we need to avoid keeping them in captivity as long as possible. And we produced a paper, which was a pamphlet, which is in Botanic Gardens Conservation International's pamphlets, if you're interested in learning more. I'm now going to pass this on to Seana, who's going to demonstrate to you what happens when we use this zoo-based model for making management decisions in plant species.
Mahalo for that overview, Jeremie, and aloha everyone. So one of the species we used in our case study to adapt this approach with plants is none other than Brighamia insignis. Many, if not all of you, I imagine are familiar with the species. It's very popular in botanic gardens and homes all over the world. The Hawaiian names include Ālula, Ōlulu, or Puʻala. It is historically endemic to Kauai and Niihau. Sadly, this species was confirmed to be extinct in the wild after numerous drone surveys in 2020 found that the last known individual on the Na Pali coast of Kauai was no longer there. Despite it being extinct in the wild, this species is extremely prevalent in botanic gardens and homes, as I mentioned. This is a screenshot I took many years ago of a video of Plant Planet owner in the Netherlands, where they cultivated Brighamia insignis by the hundreds of thousands and sold them by the botanically inaccurate name, the Hawaiian palm. The fact that all of these cultivated plants of this extinct in the wild species exist in botanic gardens made it a perfect species to do molecular genetic work, since provenance data in most cases was non-existent, and test the zoo's studbook and genetic analysis software for use with plants. Jordan Wood, for his master's project in the Plant Biology and Conservation program at Northwestern University, tested the use of the zoo's approach with Brighamia insignis. And the first step was to track founders, and by doing this, build a pedigree or stud book of all known plants. Here is a visualization of the pedigree of accessions represented at the time as live plants at NTBG's in NTBG's collection. The boxes in orange represent accessions for which there existed live plants, and that number L, I know it's pretty small, but it indicates the number of extant plants. of that accession at the time. The boxes in blue represent accessions that didn't have any live plants, so we called it dead. Each column represents a generation, and the lines and crosses are indicating crosses or selves that led to those subsequent generations. And so looking at this pedigree, we see that there are 15 founder accessions in this first column. We also see there is limited representation from the populations that existed in the southeast region of the island at Mount Ho'opu. And we see also that some accessions and lineages have been lost over multiple generations. So again, those are the ones represented in blue. The next step was to calculate relatedness between all individuals. For Brighamia insignis, we used genetic markers to identify individuals that held unique genetic diversity, since, again, provenance data was missing in most cases. We did this for collections all over the world, not just NTBG's collection. And so the main reason we did this was to see if some collections maintain diversity underrepresented in NTBG's collection that could be used in a potential future breeding program. And so here we present on the bottom of the slide structure diagrams to visualize the way that 3 different genetic clusters grouped in botanic gardens across Europe on the far left, in collections across US, and then within NTBG's collection. This data presented here was using microsatellites that Jordan Wood did as part of his master's, but we also obtained very similar results using intersimple sequence repeats back in 2014 as part of my master's work, and that was in collaboration with Jeremie. Our results indicate that there is variation among collections, and the variation among NTBG accessions does not reflect the extent of this total variation that exists. So there's value in material being held in different collections, which resulted in maintenance of these different genetic clusters. And here are some summary statistics for the diversity of the ex situ population. And we're comparing pedigree data only and genotypic data. The important takeaway from this is that we got different numbers based on the type of data we used. For example, gene diversity was predicted to be higher with the pedigree data only compared to the genotypic data, and the mean inbreeding was much higher when using the genotypic data than the pedigree data. We expected this to be the case for Brighamia insignis, that pedigree-only data would overestimate the diversity being maintained. The third step was to import the genetic data into PMX and use it to make management decisions. PMX has dozens of features, so I'm just going to go over a few of the resultant features that we found to be the most interesting and useful, such as identifying genetically unique and hence valuable collections and individuals, identifying best breeding pairs to minimize inbreeding, and to determine lifespan in ex situ collections. For instance, this is in comparison to NTBG's collection, but these 5 collections that I've written out here on the left have individuals that make them the most genetically valuable. 1 is in Hawaii, 2 are in the continental US, and 2 are in Europe. One of the most useful features is the ability to determine the best breeding pairs. So PMX showed that the best pair consists of 1 plant from the Waimea Arboretum and 1 plant from a botanic garden in Switzerland. These breeding pairs were selected based on their low level of kinship and the low level of inbreeding that their potential offspring would have. And so this is really, these examples are really showcasing the importance of coordinated efforts to conserve our living collections of rare plant species across institutions. Across collections, we were able to see that around 50% of plants live about 8 years, and most plants in cultivation do not live past 23 years. Now I'm going to share some work we did recently based on coordinated crosses that were conducted at Chicago Botanic Garden in 2016. We generated progeny of Brighamia insignis, curated there, that represent 5 cross types, including a self within Chicago's original collection. And the 4 pollen donors were represented by 3 different institutions. 1 was from NTBG, 1 from San Diego Zoo, and 2 different accessions from US Botanic Garden. This is Andrea Kramer at Chicago Botanic Garden conducting the crosses. Fruit was collected once mature. Seeds representing those 5 different crosses were all sown on the same day in 2016, so plants are all the same age. And so we compared the fitness of progeny, including plant size and pollen production and viability. Here is current master's student at the Northwestern University, the program in plant biology and conservation, Jeremie Fant, collecting pollen for these measurements. We genotyped the progeny and parental accessions to measure changes in diversity and relatedness. Genetic diversity was measured using expected heterozygosity and percent polymorphic loci. Just a note on the asterisks by the Chicago Botanic Garden and NTBG cross. Genetic data, so this high relatedness value of 0.47, for example, suggests the progeny from this cross is likely a self and not the result of a cross with the intended NTBG donor. One of the reasons we were interested in doing this small study is because we've noticed the vast majority of plants in NTBG's collection have not been producing pollen or producing only very small amounts that is mostly non-viable over the years. And we were curious whether this could be due to inbreeding, so wanted to examine whether increasing genetic diversity through strategic crosses would result in more vigorous plants that produce more and more viable pollen. And so we found that plant height was significantly higher in progeny derived from the San Diego Zoo cross compared to the other progeny. In this and the other figures, I'm going to show you the cross type is ordered on the x-axis from progeny with lowest to highest expected heterozygosity, which is one measure of how genetically diverse they are. And here's a photo of randomly selected plants representing each cross type to also illustrate what I just presented in the figure. When measuring the average pollen production per flower, we found that plants from the self-pollination produced no measurable pollen. Of the progeny that did, NTBG cross, which again we suspect is a self, produced the least amount, while the San Diego Zoo progeny produced the most. Similarly, pollen viability was lowest in the progeny resulting from that attempted NTBG cross, but likely a self, around 68% viability, which was significantly lower than progeny from the other crosses. The highest percent pollen viability was in progeny from that San Diego Zoo cross at about 89%. Based on all of this, we recommend that institutions which hold exceptional species consider how diversity is maintained within their collections and conclude that implementing a pedigree-based approach to managing reproduction of plants ex situ will slow the inevitable loss of genetic diversity and in turn result in healthier collections. And I just wanted to acknowledge my co-authors on this work. We recently submitted a manuscript to a special issue on exceptional species, and also want to acknowledge the funding that has supported that work. Development of a database and population management software to enable botanic gardens to better manage exceptional species in these ways is underway. So as Jeremie mentioned, scientists from botanic gardens and zoos are working together to do all of this. This may change, but currently the project is called TREES, standing for Tools and Resources for Endangered and Exceptional Species. And the goal is to promote effective and cooperative management of threatened plant species across botanic garden collections. It is our hope that successful genetic management ex situ will lead to more resilient plants for restoration in situ for Brighamia insignis and many other plant species.
To wrap up this presentation, I'd like to talk a little bit about what is next and what are the next stages. So in doing the work with Brighamia, we tested quite a few markers that span everything from microsatellites, which is some of the work Seana has shown you. But we have even gone into genomic level data where we're using baits from Companulaceae and even the 353 baits that Norm has talked about previously. And so with this has broadened quite considerably the type of questions that we can ask and even how useful this sort of work could be for other groups. In particular, I recognize that many of the species, Brighamia is a member of a group of lobelioids in Hawaii, and many of these species in this group are looking for, need some help and are of concern. And so at a previous conference, we held a work group to sort of talk to people about what we are doing and let them know that we have these markers and that we've tested them and that we are willing to work with them if they're interested in testing how well these markers could help answer their questions. And so with that, we set up a survey. I had some help from Matt and Maggie to sort of survey everyone and work out what sort of questions they have. And I realized that, you know, at some level we're talking genetics means a lot of things to a lot of people. And so the questions span quite a spectrum. And so as we've seen from this workshop, there are a lot of types of questions that people have. But broadly, I sort of broke them down into population genetic type questions, which were really about appropriateness of genetic material, how much genetic diversity we have, and those sort of questions. And then we had some questions which were really about sort of the hybrid between taxonomy and population genetics, the idea of, do I have a very unique population, or is there hybrids occurring between my individuals? And then finally, there was those taxonomic questions, which really not usually my strength, but the markers that we're developing span the whole spectrum from population genetically appropriate to taxonomically appropriate. And so we sort of started working through some of these questions and decided to start helping those people that could get the material. And so I worked with Matt and Lauren to get permits, and then people started sending us material. And some examples of the things we were working with, I'm working with Susan on Seana St. Johnii. She sent some samples of many of the populations that she does have, and including plantings that they have done on of this species. And although this work is still ongoing, we're already starting to see some results that are probably lean towards my own biases on the genetics of reintroductions. But within the samples that Susan sent us, there were 3 examples of plantings. 2 of those plantings were just plants that were taken or transposed from one source population and put out into another population. And what we saw, is quite interestingly, is already there's slightly less diversity in these plantings than was found in the source, although nothing too significant. We see that inbreeding seems to be negative, but this is actually due to the fact that in these plantings there were 2 populations mixed, and so there seems to be an assortative mating approach, but really it's just 2 very distinct populations in the same plantings, and that we see that relatedness of the plantings or individuals in the plantings are the same as what you would find in a source population. What was interesting to me was that some of those plantings were result from crosses that were done within situ. And what we're seeing from using crosses is already something that I would equate with best practices. We're seeing that by using material that is derived from crosses, we're seeing elevated levels of genetic diversity, even small but still significant, and that we're seeing that inbreeding has disappeared and relatedness is down to 0. And all of these are sort of what we would hope for in a reintroduction. Another example that working with Susan is a plant that's recently been discovered, which even with hand pollination does not seem to be producing many viable fruit. And so what we're seeing when we look at the genetic diversity of this taxa is we're seeing that its genetic diversity, although seems as good as what we're getting from Brighamia, which may not be a great thing, but still better than the situation in Brighamia, it does have very high levels of inbreeding, just like in Brighamia, and consequently is probably seeing the same reproductive declines that we may be seeing in Brighamia. And so this is something of concern, and maybe we need to change management with those sort of practices in mind. Another taxa that Susan has been interested in is Lobelia ohaensis, and in that she's noticed some morphological variation in plants in collections, which look more similar to another taxa. And so with that work, although microsatellites, I did some microsatellite work with these plants, and we do see some evidence that her instincts are right, and that the plants are potentially not distinct, and that this lobelia may actually be either a hybrid with the other species or maybe a misidentified individual. But already there seems to be suggest that what she's seeing in the field or in the greenhouses may actually represent some important differences genetically as well. Now, I will say that this microsatellites are probably not the best data set to use for this sort of thing. And so we are going to put these samples through a more genomic-based approach, a more taxonomically based approach, and to try and see if there is any evidence of hybridization. As I pointed out in the previous slide, we have been working with genomic data, and some of that genomic data we have thrown in other lobeloids in, and we can see that this genomic data is good at distinguishing taxa, and so at a taxonomic level we might be able to answer some of those questions about relatedness. I'm also working with Adam Williams. He sent some samples also of different variants of one Seana species, some of which might be thought to be a hybrid, or others that have been discovered which we think are the same as that species. Again, all of these are taxonomic questions that I'm not fully qualified, but I am going to help generate the data and work with some taxonomists to see if this data does reflect some of the suspicions that some land managers have seen themselves. So with that, if you have any questions, please do feel to reach out, or we can answer questions after this. Thank you.